Right. I will now record the lectures and post these to a YouTube channel. So, if for some reason you can't make it to the lecture component, it's not the end of the world. But do know that in this class, if you're used to my asynchronous online classes, we are going to have some just straight up discussion days. And we will also have live quizzes in here. So attendance is much more important here. Of course, attendance is not even a thing in asynchronous online courses. And for those of you that have had both of my asynchronous online classes and this, especially in the FCQs, I would be very interested to hear your opinions on which you prefer. This format, of course, I think provides a lot more personable social interaction, which I think is important for really feeling like you're part of a real class. But the asynchronous format provides a lot more flexibility. So I can see benefits of both. So I would just be very curious to hear some personal opinions about that, later in the semester, actually, after you have a good sample of, this versus that. Today, we're primarily going to get through lecture, though, and then, of course, eventually at the end of the semester, we're going to turn more and more into a discussion-based course, and that's where I think this is really going to get fun when we get to the Sapolsky book and really start chatting about that as a group. But to get there, we are going to have to go through some pretty complex biology and endocrinology, so you understand sort of the base, physiology underlying what we're going to be talking about. And that's what I'm going to get into today and the next few lectures. But I wanted to start with just a current event thing, which I also think frames a little bit and might have some relevance for the first argument paper that you have to produce, which I do think will require some very deep thinking on a very hot button topic that, I think could produce a lot of emotional reactions in addition to logical ones. But that is, of course, President Biden has signed an executive order to allow transgender people back into the military, which were temporarily banned under the previous presidential administration. I think that has direct relevance to this course, of course, and I guess in reading some of the arguments pro and con on the media, I see a lot of logical fallacy. And by that, I mean that, how can one argue that, someone that's transitioning from, let's say, male to female might have a competitive advantage in female sports and therefore shouldn't be allowed to participate. However, they also shouldn't be allowed in the military because I guess that would give us a competitive advantage. I don't understand the logic there. I guess it's complicated. I mean, there's a bit more to it, of course, but that is one logical fallacy I see. The other would be, I guess, transitioning the other way, where, you'd be getting testosterone supplements that would, in theory, increase physical strength and combat performance. And then not to mention, there are a large majority of careers in the military that aren't combat related. So I guess I just don't understand the entire thing and, would, I guess, advocate for a military that is, happy and, functional. But, anyway, hot button subject. Anybody have any thoughts on that? I'm sure you do, but it is sort of a hot button subject. It's tough to share about. Feel free to email me any thoughts that you might have, or, maybe we'll even consider this in greater detail as we write the argument paper on transgender personnel.
I know an argument that I've heard specifically when talking about this is people arguing about, like, how can someone that's transitioning, like, how can they expect our tax dollars to pay for it? And an interesting rebuttal is that, like, $41.6 million a year are spent on Viagra for military. So it's like you can't with such a small percentage of people being transgender, which would reflect in the military, it's not really a logical argument there.
That's a good point. That's a very good point. I mean, that is a very common argument, the cost involved in paying for that transition, which I think just bothers certain people. And but yeah, I think that's a very good counterpoint, the Viagra point. I'd never thought about that. Thanks for sharing. Anyone else?
I'll take a crack at it for just a minute, because I actually have a transgender friend that I've known for quite a while in the military.
Statistically,
And
it would be in the class as well. But please go ahead.
yeah, and yeah, I think, you know, there's honestly just no perfect answer for it. And of course, I have a bias for keeping him in the military. I think he's an amazing service member, amazing officer, and I've known him since he was enlisted. And I was his direct supervisor before he transitioned. And that person has an amazing set of qualifications to be in the military. And are there struggles within that? Yeah, absolutely. But there's also equal struggles for women. There's also equal struggles for minorities. Those are always going to be apparent. And so as long as the individual, I think, meets criteria that are morally good for the group, I think it's morally good for society in the end. And that's a really complicated idea to even pull apart, but that's kind of the basis of what I think.
Very good thoughts. I mean, you know, yeah, I also have my brother-in-law is transgender, and you know, I guess he, you know, since he came out, he was originally an aunt, and now he is an uncle. So yeah, but he prefers to go by Knuckle. That's what my kids call him, my uncle and my knuckle. But yeah, anyway, yeah, like it's you know, a very you know, interesting and important and very relevant and timely consideration. I mean, like, you know, we're seeing a lot of debate about this in Congress and things flipping back and forth at the presidential level. You know, yeah, I definitely a great thing to keep thinking about, you know, as we progress through the course. But I guess let's go ahead and jump into the lecture here. Let's see, maximize this. All right, can everybody see that? And how is the lighting? Is my lighting okay? You can see my face at least. All right. Yeah, I guess it sort of has a spotlight type effect with the lighting. It's something I've been playing around with a lot, the lighting, you know, in these virtual meetings. And I can't decide exactly what's right, but I guess this will do for now. So today, we are primarily going to get into some of the basis of endocrine function. There we go. So first, I mean, a little review. Can anybody describe the first formal endocrinology experiment that we described last time, the Berthold?
Was it the one with the roosters?
Yes. Exactly, the roosters. And then what did he find? I mean, like, so, and what did he use as a behavioral dependent measure? Like, what were his dependent measures resulting from transplantation of the testes from one rooster to another, castration early in development,
I want to say it was like the aggression levels.
absolutely. Aggression was one of the behavioral outputs, in addition to basically mating behavior associated with the hens. You know, if you castrated the animal early in development, you know, you would eliminate, you know, the sort of the sexual and aggressive behavior associated with the testes. You know, so that was really one of the first experiments demonstrating that, you know, there is this sort of molecule, this sort of chemical substance that, you know, is not in the brain, that might affect the brain, but to influence behavior in addition to other physiological manifestations. So, here's just a brief outline of today. First, I'll give you an overview of hormonal communication, and then I really want us to think about behavior and how we measure behavior and how we determine if a hormone is related to behavior. We're also going to go through quite a few basic definitions, and I'll give you a more detailed overview of hormonal communication. Specifically, though, I like to be very, very specific here on the things that I think are the most important, which are also the things that I quiz you on. I'm not going to go dig out esoteric information from the book to trick you. I want you to know, especially these things from the lecture. So first, what are the criteria to establish a causal effect of a hormone on behavior? You need to be able to diagram simple positive and negative feedback loops at 3 levels. For example, the hypothalamus, the pituitary, and then some other endocrine system in the body. I would like you to compare and contrast neural versus hormonal signaling, and to be able to compare and contrast the 3 mechanisms for why hormones will be released. In the process of this lecture, you're going to hear a lot of jargon, a lot of technical terminology, which is somewhat unavoidable. So what I don't really want you to do is feel the need to memorize every specific thing, like you might have to do in, for example, human anatomy, because that's what human anatomy is. Here, I care more about sort of the physiology, the big picture, how these things function. I mean, can you list the hormones related with every system in the body, but rather, can you diagram a system that you think is a good example of hormonal function? So let's start at the beginning. What is a hormone? A hormone is an organic chemical message produced and released from specialized glands called endocrine glands. Oh, and I also wanted to point out one thing. I try to keep track of the time here, and I know the class ends at 10:45. I totally see myself just blanking on that. I have a space cadet gene, and I'm just going to go over that. So if I hit 10:45 and I'm still lecturing, somebody stop me. I will not be at all offended if you remind me of the time. So the hormones are organic chemical messengers. They come in different, chemical structures, versus, like, for example, a steroid-based, lipid-based, peptide-based, and we'll get into some of those distinctions here, in a little bit. Endocrine glands synthesize and store hormones, and these are ductless glands, not like a mammary body. But these glands have a sensing system and signaling system which regulate the duration and magnitude of hormonal release through feedback from the target cell. This sort of feedback is just a key and central phenomenon to hormonal regulation. As I want you to get the picture that hormonal regulation is really all about balance. Like too much or too little can be bad. So, we really have these, it's sort of like a thermostat in a way, when it gets too hot, it shuts off. When it gets too cold, it turns on. That is essentially the basic concept of how a hormonal system works. The endocrine system is distinct from the nervous system, in a variety of ways. They are both major communication systems in the body, and they do work closely together. Pretty much the majority of endocrine functions that affect behavior do so through interaction with the nervous system. They integrate stimuli and respond to changes in external and internal environments, and both are crucial to coordinate functions of highly differentiated cells, tissues, and organs. But unlike the nervous system, the endocrine system is anatomically discontinuous. You have your brain and your spinal cord, which I'm sure you've all seen, in numerous classes and probably dissections, but the endocrine glands are, dispersed throughout the body. One of the big differences is that the nervous system exerts point-to-point control through nerves, very similar to sending messages by a conventional telephone. If you guys remember those, we used to have these things called telephones that would hang on the wall or something like this. But the nervous system is controlled through electrical impulses. And by nature, it's a very fast and sometimes short-lived communication. The endocrine system broadcasts hormonal messages to essentially all cells by secretion into the blood and your extracellular fluid. So it's a lot more like a radio broadcast than a telephone. It requires a receiver to get the message as well. And that's very important. I mean, I cannot overemphasize the importance of the receptors. You can increase or decrease hormones, but I mean, if there's not a target receptor, you aren't going to get an effect. In the case of the endocrine messages, cells must bear a receptor for the hormone being broadcast in order to respond. Here are just two examples, that depict neuronal versus hormonal communication. I think one of the big differences that you can see is that the hormones are released directly into the blood, where they would, spread throughout the body to the effector, organs or cells. Whereas in neural transmission, you are the neurotransmitter chemically is often released into a synaptic cleft, where it would act at postsynaptic receptors. The synaptic cleft, one thing that you might not have gone over completely in biobasis is that in the synaptic cleft, and it's not really shown in this diagram, the uptake transporters are typically located at the edges of the synaptic cleft. So they're there to prevent significant spillover outside of the synaptic cleft. Diffusion does occur outside of the synaptic cleft. It's something that I chemically detect. I mean, when I measure a neurochemical, I'm not in a single synapse. I am measuring diffusion outside of it. But there is a significant level of transporter right at the periphery of that synaptic cleft that prevents its release into a greater environment. So it's much more regulated. And then not to mention, there are glial cells that can take up excess transporter, and many enzymes that can degrade the transmitter, very rapidly. So, very distinct differences. And one other question, or one thing to consider, is which one do you have more voluntary control over? Right now, I want you to try to increase your rate of breathing. So what's happening there is your pons, so your medulla typically has just a regular steady state signal to your diaphragm, causing you to breathe passively. When you want to actively breathe, your pons sends additional neural input to muscles around your lungs, which can increase your rate of breathing in addition to your diaphragm itself. So, you seem to have some control at some level of your neurotransmission. But now I want you to increase your thyroid function. Go. Yeah. So, I mean, that's another big distinction. The hormonal or the endocrine system is, not something that you have as much voluntary control over. A cell is a target because it has a specific receptor for the particular hormone. If the cell is missing the target receptor, there's not going to be an effect on that cell or tissue or organ system. Most hormones circulate in the blood, coming into contact with essentially all cells in the body. However, a given hormone usually affects only a limited number of cells, which are called target cells. A target cell responds to a hormone because it bears receptors for that hormone. So if there's no receptor, it doesn't matter how much hormone is present. Simply, no receptor equals no effect. Let's repeat this because it is important. I mean, basically, if there is no receptor, it doesn't matter how much hormone is present. If there's no receptor, there's no effect. And I will add that there is a, arguably a distinct little caveat here in that there are non-canonical ways that hormones can regulate neural activity, which we're going to get into here, but it still involves receptor targets. And that is, where you have your traditional stereotypical receptor on the plasma membrane. That's what I'm sure what everybody has in their mind right now, hopefully at least. But there are also cases where the hormone can, depending on the hormone, can transverse the plasma membrane and act intracellularly, but it would still have receptor targets within the cell. So there are several principal functions of the endocrine system. These are the maintenance of the internal environment, maintaining the optimal biochemical environment. And again, it's all about balance and regulation through these positive and negative feedback loops. That's the big picture. That's what I really want you to get, more than memorizing like every little term in the book. It's also involved in integration and regulation of growth and development. I mean, like, like Big Mouth, the TV show, or puberty, that's hormones. I mean, like, as you obviously know, but I mean, like, basically gonadotropic releasing hormone isn't released when you're young. And as you mature and hit puberty, you get a lot more gonadotropic releasing hormone, which would then activate receptors in certain brain areas of the body to induce puberty. But, there's also control, maintenance, and instigation of sexual reproduction, including gametogenesis, coitus, fertilization, fetal growth, and development and nourishment of the newborn. We'll go over some great examples there of negative and positive feedback, but, right. I mean, one example is that, for example, milk production, is tightly regulated and, is subject to positive feedback. If the, infant is suckling around the alveolar area, that leads to milk letdown and feeding for the newborn via oxytocin, actually. But anyway, we'll come back to that example. So the big picture is that there's coordination of physiology and behavior of an animal by regulating, integrating, and controlling its bodily function through very critical balances. And some of these balances are cyclical. Some of them have diurnal variation, like night versus day. Some of them come on at certain stages of life and by certain triggers. The interaction of a hormone with its receptor is also important to consider. What actually happens when a receptor is activated by a hormone? Well, if you've taken intro to biology, this is probably very familiar, but you essentially start an intracellular signaling cascade of events that is really multifaceted. I mean, measuring, like just diagramming, like, I don't know, some kinase activates, Krebs activates, calcium, IP3 channels, blah, blah, blah. But there are literally dozens, if not hundreds and thousands of these intracellular signaling cascades that are all interacting in a very complex manner. And integration occurs through, additional proteins inside the cell, like DARP32 is one, a famous one. But anyway, essentially when you bind to the receptor, that would start a series of cellular events that eventually lead to activation of either enzymatic pathways that can go on to produce lots of different chemicals that can then have their own effects, or to the effects on gene expression and protein synthesis, leading to, for example, upregulation or downregulation of receptors expressed in the plasma membrane. So Berthold's secretory blood-borne product is now known as a hormone. Hormones are, again, these organic chemical messages that are produced and released by specialized glands called endocrine glands. The endocrine system is, unique from duct systems like saliva glands and mammary glands, where the substance that would be secreted is contained with or released through a duct. But hormones are these messenger molecules that are released through ductless glands into the blood. where they act on then distinct target cells that have receptors for them. Target cells respond to the hormones, again, if they have the appropriate receptor, and the effects are dependent on the program response of the target cell, which would really be determined by the intracellular signaling components within that particular cell that would have been selected over many centuries through natural selection. Hormones are really just molecular triggers, trigger these intracellular and molecular cascade events. So hormones coordinate the physiology and behavior of an animal by regulating, integrating, and controlling its bodily functions. One example would be, the same hormone, luteinizing hormone, that would cause egg or sperm maturation, also stimulates mating behavior in many species. And I mean, this is developmentally important. So essentially you wouldn't have or observe mating behavior before you had the sperm and egg, associated with the end product of mating for biological purposes, for natural selection. So, I mean, essentially that is why, in theory, those behaviors and chemicals were selected in a developmentally regulated manner. Hormones are similar to neurotransmitters, but can operate over a greater distance and over a much greater temporal range than neurotransmitters because they're released into the blood. There are several important distinctions between hormones and neurotransmitters. Neural messages can only travel along existing nerve tracts. Hormonal messages can travel through the circulatory system, thus any cell receiving blood is potentially able to receive a hormonal message. Neural messages are digital is one way of thinking about it, if you're familiar at all with physics or electronics. But digital messages are all or nothing events that have a rapid onset and offset. However, hormonal messages are more analog. They're graded events that can take seconds, minutes, or hours to occur. How does a hormone exert its influence? Only target cells, again, with receptors for that hormone can be influenced. There is an interaction of a hormone with its receptor that leads to a genomic response whereby the hormone activates genes that regulate protein synthesis. For example, upregulation, synthesis of a receptor for that hormone, etc. And this is typically done through regulatory proteins intracellularly. And of course, at some level would involve, transcription and translation, the nucleus, the endoplasmic reticulum, et cetera, et cetera. But some hormone effects are non-genomic, and this is, important to understand. Non-genomic basically means transcription independent. There were effects that would have a rapid onset of action because you're not actually waiting for that sort of long-term molecular change of transcription and translation and increases in receptor number, which might be longer-term effects. But anyway, these non-genomic effects are rapid and likely not mediated through intracellular receptors, there still would be intracellular cascades, but not necessarily the intracellular receptor. The non-genomic action would include secondary messengers like cyclic AMP, diacylglycerol lipase. Interestingly, if you are curious, diacylglycerol lipase, it's actually an enzyme that produces many things, one of which is endocannabinoids. So your brain's natural cannabinoids are produced by diacylglycerol lipase. But anyway, there are also many different kinases, which are mitogen activated, like one would be mitogen activated protein, and ion fluxes, like just changes in calcium ions alone can induce a big series of events, really can profoundly alter, transcription, translation, in addition to a variety of other ultimate effects. estrogen is one example that acts both genomically and non-genomically, whereas, for example, a monoamine like dopamine would only act non-genomically. It's not going to, pass into the intracellular space and act on, targets intracellularly. It's important to note that sufficient numbers of receptors must be available for a hormonal effect to occur. It's a popular belief that individual differences in behavior reflect differences in hormonal concentration, which is not necessarily true a lot, because it really depends a lot on the receptor. And the individual differences in behavior as well can reflect many things. It's not necessarily just high testosterone, for example. I think we oftentimes jump to too many conclusions about hormones in this way. It's just something that, I think is probably a little inappropriate, and sometimes everybody, including myself, is probably guilty sometimes to jump to too many conclusions about hormones when really the, total effects on a behavior are really just a complex cacophony of hormonal, neuronal, and really immune, gastrointestinal, function. But hormones rarely change the function of a cell. Rather, they alter the rate of normal cellular function. Thus, hormones would affect cell morphology, meaning like basically its shape and size. It would also influence the development of muscles and neurons, and affect cell death, apoptosis throughout the nervous system. One example there is testosterone. I mean, if you read the literature, you're going to find a whole ton of different conflicting things about testosterone. You're going to find some that say it's somewhat protective, but you're also going to find some that report that increased testosterone can lead to apoptosis or cell death. But I mean, again, this is hormones and behavior, not just basic endocrinology. So, how the hormones actually affect behavior. And before we get into that, we should think of what is behavior. Behavior, in a scientific perspective, is, an output. Essentially, that there is like some sort of behavioral output that you can measure, that you can quantify. So how might behavior affect hormones? Behavior can and often does affect hormonal levels, which in turn can influence subsequent behavior. One example here is, what goes on during a sport match. I love this example, as an avid sports fan. But, in a study, scientists measured testosterone levels of fans in the World Cup championship between Brazil and Italy. Brazil ultimately won on penalty kicks, and when they measured the fans afterwards, the Brazilian fans showed an increase in testosterone, whereas the Italian fans whose team lost, showed a decrease in testosterone, which, it makes you want to have all these sort of, assumptions about what goes on afterwards and hooliganism and this and that. But, again, such things are complicated, and it's not just like a simple relationship there. There are many things going on to the losing side that could, spur aggression as well. So behavior can and often also affects hormonal levels, which in turn can influence subsequent behavior. For example, in one study, the investigators measured testosterone levels of 4 heterosexual couples over a total of 22 evenings, and they took multiple samples, some before and some after sexual intercourse. And, what they found was that basically testosterone increased after sex in both men and women, No changes, and also no changes were observed on sex nights. But in both women after having sex, you observed an increase in testosterone. In addition, they looked at early evening samples, which showed that there was no difference in testosterone concentrations between the sex and no sex evening, suggesting that in this case, sex increased testosterone more than testosterone led to sex. And another, confounding variable here is just the concept of physical exercise. So was it actually the sex that was increasing testosterone levels, or was it physical exercise, which can also impact testosterone? It's also important to consider confounds, in these sorts of assays. I mean, especially with behavioral outputs. I'm a behaviorist. I'm a behaviorist, a pharmacologist, and a neuroscientist. I love the study of behavior, but we sometimes, get dumped on a little bit, quite frankly, by, for example, a molecular biologist who would study a very consistent change in intracellular signaling. It's going to be incredibly reliable. There's more noise in behavioral assays. I mean, there's more different things that can affect or influence your behavioral outcome. And, I don't really think that means that behavioral assays are bad. It just means they're complicated. And that's something to keep in mind when considering the outputs of these studies. And again, I love behavioral assays. I am a behaviorist. The study of the hormone-behavior relationship is organized around the idea that animals are composed of really three interacting components. This is really just a big picture. There's an input sensory signal, some sort of integration, which is almost always the brain, and then an output system, which is your muscles. This is essentially the basis of the majority of behavior. I mean, there are simple reflexes in your body that can occur through still a nervous system, but outside of your brain. And, lower animals, like for example, the lobster, lacks a brain, and they have a diffuse motor system, which also, brings up the idea, what does a lobster feel versus a human feel, for example, when, being cooked? It would be incredibly, incredibly different. And it's hard to anthropomorphize and say what that lobster is feeling, but without a central nervous system integrator, I would have to expect it would be incredibly a different experience. But anyway, almost all of our behavior, aside from simple reflexes, involves the brain as an integrative system, and then action. Most of our, would involve our muscular system, which receives neural input. One example here would be removing the testes of male zebra finch stops its singing. And then if you reimplant the testes or provide the testosterone, and the singing resumes. So, this obviously shows that testosterone is involved in singing, but it doesn't really explain how. And there are multiple possibilities here that I would like you to consider. First, there could be a change in the actual sensory system. Does testosterone alter the bird's sensory capabilities, making the environmental cues that elicit singing more noticeable and more salient? If this were the case, females or intruders might be seen or heard more easily. In the central nervous system, could this be affected? Testosterone could change the neural architecture or the speed of neural processing. And higher processes like motivation or attention might also be affected. Finally, there are effectors. Here testosterone concentrations might affect the muscles of the syrinx, which is the avian vocal cord. This three-part framework can aid in the design of hypotheses and experiments to help understand how hormones affect behavior. It's something you would have to consider at what level is the hormone actually influencing behavior, possibly more than one. So here's just a simple diagram, showing the relationship here. And, it's again important to note the reciprocity here. Behavior can affect hormones. Hormones can affect behavior. And it can do so, by really interacting with the hormones' influences on the central nervous system, the central processor, which could then influence both sensory systems and effector systems. But hormones are also acting on these systems directly, as is behavior. So it can become very complex, to consider. But the key thing that I want you to take away from this is how reciprocal these regulatory mechanisms are. That behavior can affect hormone, hormone can affect behavior, really through a variety of ways. And really, the brain ultimately is going to come back and be your central processor. This is, I think, an important concept to consider as well. So, I mean, how do you really establish a causal link between a hormone and a behavior? It's really difficult. One perspective is that three conditions must be satisfied by experimental results to establish a causal link. One would be a hormonal dependent behavior should disappear if the hormone is removed or the action of the hormones are blocked. Two would be if the behavior stops, replacement of the hormone will restart the behavior. So Berthold's roosters fit both of those two criteria. Three would be hormone concentration should be directly related to the behavioral effect. This would mean that higher concentrations of the hormone would result in a greater display of behavior. And number three is particularly tricky when you think about this experimentally, really because hormonal signaling can be very long-lasting and can occur in these sorts of, chronological or temporally regulated bursts. So when do you measure the hormone? You really have to know a lot about the neural activity of the hormonal system and how it is actually being released. And how do you measure that in people? That becomes also very difficult. So really in people, you're sort of stuck with just measuring blood content of these things, which is difficult, and I don't know that it's always going to give you a clear picture of three even, and it definitely isn't going to touch on 1 and 2. So, that is, I think, an important reason to continue to advocate for animal experimentation to some extent. I mean, otherwise, I don't really see how we can understand how the endocrine system controls behavior without being able, at this point, at least, to do more invasive, experiments. But going back to this third class of evidence, it's really, as I was saying, difficult to obtain because many hormones have long latency of action and a long offset of latency and are released in this sort of pulsatile manner that can be set to a clock, for example, or other environmental stimuli. Another problem is that biologically effective amounts of a hormone are tiny. A little drop in the blood can have incredible influences on behavior throughout due to receptor sensitivity. Therefore, it can be difficult to measure accurately. Just taking one blood sample at one point in time isn't going to tell you very much. And I mean, what are you really going to do? Basically leave a person tethered to an IV line, taking their blood every 2 minutes? And even 2 minutes might be too long. Maybe you might, it's not like you're getting a real-time blood sample here from the people. So, effective concentrations are also measured in micrograms, nanograms, and picograms. It's very hard to detect. And, also, as I was saying before, the first two classes of evidence, are considered to be more reliable, but almost impossible to do ethically in people. So, a lot would have to be inferred about people based on animal experimentation, right? Like, for example, think about doing Berthold's experiment on people. Obviously that is not, going to be a very welcomed idea. It conjures up memories of Nazism and, the worst parts of American history as well. I mean, experimenting on, minority populations, prisoners, these things have been done. And I think through hindsight, we can now see they were ethically despicable. But anyway. The endocrine system is just one example of chemical communication, which is a ubiquitous feature of life on Earth. I mean, it's essentially the natural selection that led to our basically processing throughout all, species. And really, like, basically, it just becomes more complicated, and not necessarily even better, but more complex, throughout the steps of evolution. But, this chemical mediation is conducted by several interacting physiological systems and the key components, especially for this class, the endocrine system. There are different types of basically chemical signaling that you should be aware of. First, there's intracrine, which means chemicals that regulate intracellular events. A chemical would be released and then act at receptor targets within the cell. Autocrine would be chemical feedback onto the same cell that secreted them. So the big difference here is that the chemical would be released extracellularly and then act on receptors within that same cell. Paracrine would be chemicals that affect adjacent cells or cells, very close to it. Endocrine would be an example of a chemical that's secreted into the bloodstream to travel to distant targets, obviously pretty important for this class. There are also ectocrine mechanisms. Ectocrine would be a chemical that would be released into an environment by individuals to communicate with others. Pheromones are a great example. We'll talk about this a little bit as we go on. We just got a new cat, a third cat, and one of my existing cats doesn't love it so much. So we are actually, treating the house and the cat with a pheromone that is supposed to basically be like oxytocin to some extent, and facilitate bonding. I'd say it's working okay. Again, these things are very difficult to regulate temporally in a manner that would be effective on behavior. Another one to think of is autocrine. Autocrine would fall under the ectocrine label. Autocrine is similar to pheromone function, in that, basically it's one organism releasing and affecting another, but in this case, it commonly would involve a plant. So, for example, rather, a plant releasing a certain chemical to attract or repel, an animal or insect, would be a form of autocrine communication. Here are just some diagrams of these different forms of signaling. Autocrine signaling, of course, would be the neurotransmitter being released and acting back onto the cell. This would be contrasted to endocrine signaling, where the steroid hormone would act, for example, intracellularly, and then there would be an intracellular signaling event. Paracrine would be when the released chemical acts on a very adjacent or nearby target cell. And endocrine is what we're really talking about here, where you're going to see a release of the chemical into the blood that would then travel throughout the body to act on distant target cells. A chemical message is any substance that is produced by a cell that affects the function of another cell. And you might know that chemical signaling was really sort of the entire concept of it is really associated with the founding of the field of neuroscience. You probably know the name Camillo Golgi from Golgi apparatus. Maybe you know the fish song, maybe you know the biology, but you've probably at least heard Golgi. Golgi was a very famous scientist who developed the Golgi stain, which led people to study cells. And Golgi also studied neural cells and became convinced that they were all connected and communicate through electricity as a neuronal net. It's basically all contiguous. And then a young Portuguese scientist challenged Golgi on this concept, and actually used the Golgi stain to conclude that the nervous system is really primarily consists of discrete cells that likely interact through some other molecule, like a chemical message. They actually got into a huge fight about this that really led, in many ways, to the founding of the field of neuroscience, the study of the brain. And Golgi was actually wrong in this case. Ramon E. Cajal was the Portuguese scientist that really argued that it's composed of discrete cells that have to have some chemical messenger. There are gap junctions that do allow direct electrical, signaling between neural cells. But for the most part, we rely heavily on these chemical messengers. Cytokines are a chemical messenger that evokes proliferation of other cells, especially in the immune system. And there's great interaction between the immune system and the endocrine system and immune system and the nervous system directly. Hormones would be a chemical messenger that is released into the bloodstream or tissue fluid and affect the function of target cells some distance from the source. Now, there are also some additional chemical vocabulary terms that you're going to run into the book. I just want to give you a brief overview of these. A neurohormone would be a hormone that's produced by a nervous cell, a neuron. A neuropeptide is a peptide hormone produced by a neuron. And a peptide is essentially a short chain of amino acids, maybe like a baby protein. A neurosteroid would be a steroid hormone produced by a neuron. Here you're going to see a distinct organic chemical structure, often based on cholesterol, sort of a ring structure, rather than like a little chain of amino acids. A neuromodulator, and those would also have different properties associated with a plasma membrane, associated with water, et cetera, et cetera. And then you have neuromodulators, which are a hormone that changes the response of the neuron to some other factor. And a neurotransmitter, a chemical message that acts across the neural synapse. So those are distinctions. Many of these chemical communication systems have been studied independently by separate groups of specialists. This is sort of the tendency of science, myself included. We tend to be reductionistic. We like to study the system in isolation because, of course, this becomes very complicated and there's a lot of integration. And it's becoming increasingly obvious that the nervous, endocrine, and immune systems interact in some significant manner that is very difficult to study, at a larger level. It's sort of my opinion that, I think that me and, basically most scientists right now are sort of developing puzzle pieces, in sort of these pieces in isolation. And I think that, over the next couple of generations, we're going to move away more from animal experimentation, like where we're measuring these specific, parts, and, as, computer and mathematical and linear algebra capabilities, increase, we are going to see more computational science sort of come together and put the puzzle pieces together and give us a better understanding of how these things interact. I don't know how we can do this quite yet in a dish with an electrode, but again, that's the only way that we can create the puzzle pieces. So that's sort of like the big picture of how I see all of this sort of coming together over the next few generations. But, that's just one prediction, an educated guess, if you will. Endocrinology is the scientific study of the endocrine glands and their associated hormones. Neuroendocrinology is the scientific study of a transduction of a neural signal into a hormonal signal, as well as the relationships between the nervous and endocrine system. The endocrine system, again, is a system of ductless glands that secrete hormones. Hormones are these messenger molecules that circulate in the blood and act on distant target cells. Target cells respond to the hormones for which they have receptors. The effects are dependent on the programmed response of the target cells. Hormones are just molecular triggers. The endocrine system, its general functions, or features, are that it is a system of, again, ductless glands that secrete hormones. There are endocrine glands that have a rich blood supply. They're highly vascularized, which makes sense seeing how they're receiving signal through the blood. Hormones are secreted into the bloodstream, and hormones can travel in the blood to virtually every cell in the body and can thus potentially interact with any cell that has appropriate receptors. Again, think of like an analog radio signal. Hormone receptors are specific binding have specific binding sites that interact with a particular hormone. No receptor, no effect. There are other sorts of exocrine cells, mainly salivary, sweat, and mammary glands that secrete products into ducts that then carry them to adjacent organs or external environments. This is, of course, can be compared and contrasted to endocrine release or endocrine function. One of the big differences there is just anatomical, that exocrine glands like salivary, mammary, and sweat have ducts. Water-soluble or small peptide hormones tend to be packaged in vesicles in endocrine cells. The vesicles then fuse their membrane with the cellular membrane to release the water-soluble or small peptides into the blood. Here, the water-soluble ones are unique in that they would have to then bind to a carrier protein that would carry them through the blood. Whereas if you, well, that's actually not true with the water-soluble ones. If you're insoluble molecule would have to then bind to a carrier protein. Water-soluble things like a small peptide would not need to bind to a carrier protein. They can then diffuse throughout the body, through the blood. But in contrast, if you have these sort of lipid-soluble or water-insoluble molecules, like a steroid hormone, for example, this can easily move through the phospholipid membrane. Its release is a little simpler. It wouldn't have to be packaged in the vesicle and released through that manner. However, these cannot be diffused through the blood by themselves, so they would have to bind to a carrier protein, which would then disperse throughout the system. There are many different carrier proteins for many different, chemicals. For example, glucose. Really your main energy source has specific carrier proteins that carry it through the body. I think they're called like GLUT2 or something like that. But there are also several major endocrine structures that you're going to hear about. So I at least want to go over these. And again, these are distributed throughout the body rather than being contained in one central place like the central nervous system or brain. The hypothalamus is at the base of the brain. It controls many different hormones through its actions on the pituitary gland. We're going to go over that quite a bit in the future, but there's an anterior and posterior pituitary that have very unique functions. The anterior pituitary is involved in secreting hormones by the thyroid, adrenal cortex, and gonads, and is involved in growth amongst many other things, whereas the posterior pituitary is very much involved in osmotic balance, and salt balance. The pineal gland is, involved in reproduction and also body rhythms, especially diurnal rhythms in other animals like birds. The thyroid gland is important for growth and development and metabolic rate. The adrenal glands are involved in emotional arousal, and there are distinct hormones released from the adrenal cortex and the adrenal medulla. The pancreas is very involved in sugar metabolism, as you know if you've known any diabetics, type 1 diabetics. In particular, the gut is involved in digestion and appetite control, and the gonads are involved in body development and reproduction. There are unique chemical structures of hormones. Peptides and proteins is one. Most hormones are either peptides or proteins, and again, peptides are just sort of short amino acid sequences. I think of them as basically baby proteins. There are also amines. Amine hormones are derivatives of the amino acid tyrosine, which is also the principal derivative of the monoamines like dopamine. Lipids and steroids are steroid hormones that are produced by the adrenal cortex and the gonads. Generally, one gland releases one class of hormones, and that doesn't mean one hormone, but one type. For example, the pituitary tends to release peptides that have shorter acting type regulation, whereas the adrenal tends to release steroid hormones, which have a longer lasting effect and prolonged action throughout the body. Again, we've already talked about the hypothalamus, which is essentially at the base of the brain and directly controls the pituitary, which then releases many different, hormones, which would have actions throughout the endocrine system. The thyroid gland is an H-shaped organ located on the upper trachea. The thymus is behind the sternum between the lungs. The pancreas is behind the stomach and liver. The adrenal glands are bilateral organs on the tops of the kidneys. The pineal gland is between the telencephalon and diencephalon in the brain, and the gonads are the testes in a sac outside of the abdomen or the ovaries in the abdomen. There's structural variation of hormones from species, or across species. So, this species specification is conveyed by the exact amino acid sequence. As a result, if you injected mammalian gonadotropin releasing factor into a toad, it would not affect its reproductive function. But if you did this experiment, it would be wrong to conclude that gonadotropin releasing hormone doesn't play a role in toad reproduction. It would just mean that mammalian gonadotropin releasing hormone isn't functional cross species with a toad. So in reading, be sure to pay attention to the source of the hormone in the study. So how are hormones regulated? This is a very key concept. There's typically top-down organization of the endocrine system. The hypothalamus would produce releasing factors that stimulate production of the anterior pituitary hormone, which would then act on peripheral endocrine glands to stimulate release of a third hormone. I'm going to give you specific examples of this that I would like you to be able to describe and illustrate to me. A great one to practice on a blank sheet of paper or a whiteboard. Again, I care more about understanding the overall process and example rather than you memorizing every single hormonal system or hormone. The posterior pituitary hormones are synthesized in neuronal cell bodies in the hypothalamus and are released via synapses into the posterior pituitary. Examples here would be oxytocin and antidiuretic hormone. Hormones can be regulated through sensing and signaling, which is a biological need. When a biological need is sensed, the endocrine system then sends out a signal to a target cell where action addresses the biological need. Key features of this stimulus response system are that there has to be a receipt of a stimulus, then there has to be synthesis and secretion of a hormone, then delivery of a hormone to a target cell, which would then evoke a targeted response, and ultimately there would be degradation, typically enzymatic degradation of the hormone. The physiological effects of hormones depend largely on their concentration in blood and the extracellular fluid. Almost inevitably, disease results when hormone concentrations are either too low or too high. Again, it's maintaining that crucial balance. It's important. This illustration just demonstrates or tries to reinforce the importance of balance. When the entire system can be knocked off kilter at multiple levels, but when you result in an imbalance, you can result in disease or alterations in development. Here is just another example of insulin and glucose, which could lead to diabetes, for example. And since we're low on time, I'm going to just sort of breeze through this, but I think this is a great example for you to review. If you want a great example of real typical effects that this imbalance can provide, like diabetes. So the concentration of hormones as seen by the target cells is really determined by three factors: the rate of productivity, the rate of delivery, and the rate of degradation or elimination. alterations of any of these can produce the imbalance. The rate of production is caused by the synthesis and secretion of hormones, which are a most highly regulated aspect of endocrine control. Such control is mediated by positive and negative feedback loops, as described here in more detail in a couple of diagrams here in the future. The rate of delivery, an example of this effect, is blood flow to a target organ or a group of target cells. A high blood flow would result in the delivery of more hormones than low blood flow. Finally, there's degradation and elimination. Like all biomolecules, there are rates of decay and also enzymes that can break down or eliminate enzymes or hormones. Shutting off secretion of a hormone that has a very short half-life causes circulating hormone concentration to plummet. But if a hormone's biological half-life is long, effective concentration persists for some time after secretion ceases. So here is sort of like an example of a very basic feedback loop here. So, this is sort of what I would like you to understand and be able to diagram, but maybe a slightly more specific real example. But in the case of negative feedback, which is most common, you would have a hormone that releases, a hormone is released by an endocrine gland where it acts on a target tissue, which would then produce a product. This product would be detected by receptors in the endocrine gland and shut down production of that hormone because it's too high. Like this is your thermostat turning off. Positive feedback would be your thermostat turning on, where an endocrine gland or some other sort of event would produce a hormone that could then act on target tissue to result in a product that would then activate the synthesis of more hormone at the endocrine level. One good example of this is the thyroid gland. I think that this is just a very simple example. It would be a good one to diagram. But the hypothalamus would release a hormone, in this case, a thyroid releasing hormone, which would then act on the pituitary to release thyroid stimulating hormone, which would then act on the thyroid gland to release T3 and T4, which would then activate receptors on the hypothalamus to shut down release of the thyroid. If this is disrupted, you would have hypothyroidism, which is pretty common. Many people have been influenced by it. Examples here would be Hillary Clinton and George H.W. Bush. But, it's also another possibility here is what's known as Graves' disease. So this is where you would actually have the immune system interacting with the thyroid system to essentially attack it and disrupt its regulatory processes, resulting also in hyperthyroidism. Missy Elliott has had Graves' disease, as have several, very famous people. So, what I really would like you to be able to diagram are sort of these simple feedback loops. Like, for example, here's one where, just a decrease in calcium in the blood can lead to the synthesis of the parathyroid factor to stimulate the parathyroid gland, to release the parathyroid hormone, which would then increase calcium in the blood, which would then have a direct, negative feedback loop on the parathyroid gland, which is typically activated or stimulated by decreasing calcium in the blood. In reality, things are much more complex. Whereas I want you to diagram simple feedback loops, I want you to know that things can be much more complex. For example, gonadotropin releasing hormone can be activated by the hypothalamus, which could then lead to luteinizing hormone being released by the anterior pituitary, which would activate the testes to increase sperm production and basically sexual arousal to some extent. Which is then going to, but there's going to be feedback here that can activate at the anterior pituitary or the hypothalamus to shut down release of either gonadotropin releasing hormone or luteinizing hormone. A great example of positive feedback is part of the milk letdown response, where the baby would stimulate receptors by suckling around the areola, which would then stimulate neural pathways to the hypothalamus, which would then lead to the production of increased oxytocin that would be released from the posterior pituitary, leading to milk letdown for the baby to feed. In reality, these things are even more complex. Obviously, I don't want you to be able to diagram this one, but in reality, things are much more complex, which is what the point is. In most cases, you're going to have examples of both positive and negative feedback and a lot of interaction at multiple levels. Part of this is because in reality, we're not studying just one system in isolation. There's input to the endocrine cells from multiple systems, including neurons, hormones, ions, just changes in calcium levels in the blood is going to have, major effects, pH changes, and also organic nutrients in the blood could all impact endocrine function. There are three mechanisms of hormonal release that I think you need to be responsible for. I would know these for the quiz. One would be humoral, in response to changing levels of ions or nutrients in the blood, like calcium ions. Neural, just direct stimulation by the nerves, and hormonal, stimulation received by other hormones. Neural, a good example here is, activation of the hypothalamus. Right. So, neural input to the hypothalamus would stimulate the synthesis and secretion of releasing factors, often also times hormones, which would then stimulate the pituitary hormone production and release. It's also important to note that these typically occur rhythmically, like there is a endogenous rhythm. They can also be entrained to external factors like the light or seasons. Response stimulus coupling enables the endocrine system to remain responsive to physiological demands and changing conditions. Secretory episodes occur within different periodicity, and pulses can be as frequent as every 5 to 10 minutes. Again, I really want to emphasize that this makes it difficult to measure these things. You're not just going to have a tap into a person's blood. So keeping in mind hormones are cyclical, and their release can be periodic. You can see how it would be very easy to get a false negative, if you're just trying to measure hormones here at different time periods. It's also important to know that there can be a great physiological impact of the pulsatile hormones release. For example, if you give gonadotropin releasing hormone once hourly, gonadotropin secretion and gonadal function are maintained normally. There's really no effect. A slower frequency does not maintain gonad function. Rather, you have to have fast or continuous infusions that inhibit gonadotropin secretion to block gonadal steroid production. So essentially, too much of it inhibits via negative feedback. It's this very sensitive balance, which is the key, big picture concept here. So that is it. I think I nailed the timing on that one. Probably will never happen again. So, we have 1 minute. Does anybody have any questions?
I have a quick question about if we're going to be able to have access to the PowerPoint slides on Canvas to go back and reference.
Some of them are available online, and what I'm going to do is I'm going to create, I'm recording this lecture, and I will post the lecture to YouTube so you can fast forward or rewind and get all of the slides through the video.
Thank you.
You're welcome. Any other questions? For the quiz, remember, focus on the learning objectives, focus on the big picture concepts. I think it can be a little overwhelming when you read the first couple chapters of the textbook. It's not my goal to have you memorize every hormone. What I want you to understand is these big pictures, be able to diagram positive and negative feedback loops. Give me the big picture concepts here that I'm trying to reinforce. All right, I guess we'll call it there. Remember, on Wednesday, it'll be another lecture sort of like this that I will also post online. And then hopefully very soon we'll make it through this slog of content and get into the fun discussion of the Sapolsky book. Have a great week.
Thank you.